In the first lecture, we looked at antibody structure and the genetics behind generating antibody diversity. In this second lecture, we're going to focus more on the applications of antibodies, and I touched on those in the first lecture. But before that, we need to understand a little bit more about the detail of antibody structure, and you'll see why that's important towards the end of the lecture. So, aims and objectives for this lecture are, first of all, to examine variability in a bit more detail at the level of these CDR loops that form the antibody combining site. Also to look at the applications of antibodies in more detail, look at antibody fragments that can be used, and how we can use antibodies in vivo, what we need to be able to do to make antibodies suitable for use as drugs. And at the end of the lecture, you should be able to describe those principles of antibody variability at that detailed structural level, discuss those applications of antibodies in vivo and in vitro, and discuss the problems of using antibodies in vivo and the methods that we can use to circumvent those problems. So, as an overview of what we're going to cover in the lecture, we're going to look at why antibodies are useful. We're going to do a brief recap of their structure and function. Then we're going to look in more detail at the structure and variability of CDRs, and then move on to their applications, starting with why antibodies make really good drugs, ways that we can use antibodies as drugs, some example applications, and finally the problems of antibody-based drugs and the methods that we use to get around those problems. So first of all, why are antibodies useful? And by that, what I really mean is, why is understanding antibodies useful? Well, obviously, understanding how antibodies work is really central to understanding the immune system and in particular the adaptive immune response. However, they are also invaluable as laboratory reagents used in purification and labelling. We'll come back to examples of all these later. They're also really valuable as biosensors, being able to measure quantities of particular compounds in blood serum, for example. But probably most importantly, at least from my perspective, and most interestingly, is their use as drugs. Approximately a third of drugs in development are now monoclonal antibodies or based on monoclonal antibodies. And in April 2018, there were 72 approved monoclonal antibody products, either in the US or Europe or both. And those products include what is currently the world's largest grossing drug, adalimumab, or also known as Humira. And that's a drug that's used for rheumatoid arthritis and is a fully human antibody. So, having just emphasized the importance of antibodies, let's go on to recap their structure and their function. So naturally, they're molecules of the adaptive immune response. They act as this interface between the almost infinite variability of antigens from bacteria, viruses, fungi, etc. They act as an interface between that and the constant effector functions of the immune system. In other words, how we're going to kill that invading organism. And the key functional aspect of their binding is that they have both high specificity and high affinity for their targets. Just to refresh the genetics very briefly, you'll recall that the structure is formed of light chain and heavy chain that come together, and we get two pairs of light and heavy chains, so that we end up with a Y-shaped molecule consisting of two heavy chains and two light chains. The light chains can either be of the lambda class or the kappa class, and their variability is formed by splicing together gene fragments. So in the case of the light chain, we have about 30 of these V fragments, which join up to one of these about 4 J fragments, or segments, and therefore you get 30 times 4 different sequences that you can create. Similarly with the kappa chain, about 40 of these V's, 5 J's, giving you about 200 different chains. And in the heavy chain, about 65 V's, 27 D's, and 8 J's. But because the D segment can go in any one of 6 reading frames, and you can get extra bases inserted before or after the D segment at random, this gives you millions of possible antibody sequences. On top of that, you have somatic hypermutation that introduces additional mutations, and there's evidence that that occurs preferentially in these CDR loops that form the binding site. So we have this huge amount of variability. From that simple domain splicing, we have about 1.3 billion different sequences and structures that we can produce. But when you take into account the somatic hypermutation as well, we probably have a complete repertoire of more than 10 to the 15. So, as I said, we want to look in a bit more detail at the structure. So zooming in then from the overall Y shape of an antibody and looking at what's known as the Fab fragment, one of these arms, which is so named because it's the antigen binding fragment. We can zoom in on that, and here in fact I've zoomed straight from that to the FV fragment. Going back to the last slide, that's the region at the tip of the antibody arms here. This is where all the variability actually is. So I've zoomed in on that, put it upright and showing its interaction with an antigen. Now, what I've done here is to take that previous picture, take away the antigen, and then roll the FV fragment towards you. So at the top of the picture comes towards you, and you're now getting an antigen's eye view of that combining site, the antigen combining site or binding site. And top right here, we have highlighted with the thicker lines these 6 CDR loops that form the combining site. And I've labelled those below with L1, L2, L3. These are the 3 loops of the light chain, H1, H2 and H3, the 3 loops of the heavy chain. And those are labelled in the sequential order that they appear in the protein sequences, working from the N terminus towards the C terminus. Now, bottom left, I have taken that same view that we had in the top right, but now I've space filled all of the CDR loops, including the side chains, rather than just showing you a C alpha trace. I'm showing you all the atoms, and you can see that they form an essentially continuous surface with a few little gaps between them where framework residues, in other words the regions that are not the CDR loops, poking up to fill those little gaps. But essentially this binding site consists of just these 6 loops. So let's have a look at the structure of the CDR loops in a bit more detail. Now you'll recall from the previous lecture that Wu and Kabat back in 1970 looked just at the sequences of antibodies and identified, by aligning those sequences, these hypervariable regions 1, 2 and 3. And based purely on that sequence information, they suggested that these would form loops and that they would end up coming together to form a binding site, and therefore they referred to them as hypervariable loops or complementarity determining regions. Now, as we've seen, their prediction was exactly right, and these 6 loops, 3 from the light chain on the right here, 3 from the heavy chain on the left, come together to form that binding site. And these loops are supported on a relatively conserved framework, and you can see that by going back to their variability plot. You can see these framework regions in between. Although they are variable, they're clearly not as variable as the CDR loops. And when we look in terms of structure, the structure of these loops is actually very conserved. So what I've done here is taken about 20 antibodies. Some of them are from mice, some of them are from humans. I've chopped off the CDR loops and then I've just fitted them to one another. And you can see that their shapes really are very, very similar. There are one or two little bits of variation. But if we look in more detail just at the heavy chains, which are shown on the left here, so this is heavy chain on the left and the light chain on the right, and I've tried to fit both together. But if I just look at the heavy chains, this is the same set of heavy chains, and you can see this in the picture on the right, that now they're clustered much more tightly. And that's because when you look at a VH and a VL domain, there is this variability in the packing between the two domains. So if you're trying to pack something which is skewed one direction with something which is skewed in the opposite direction, then you end up having to get this kind of spread of conformations. Whereas if we just focus on this region, which is what I've done on the right here, then you can see a much better fit between the two structures because we're not being kind of diverted by the need to accommodate these light chains as well. So going back to Kabat and Wu again, the assumption was that these three loops, which as I've shown here, map quite nicely onto the three loops in the structure. So we've got the red, green and cyan, red, green and cyan being L1, L2 and L3. Given this a huge variability in the sequence, there was an assumption that we would also get a huge variability in the structure. However, Cyrus Chodhia, working in Cambridge, started to look at this structural variability rather than sequence variability, and he looked at approximately 8 crystal structures that were available in the mid to late 1980s. And he found that despite this huge amount of sequence variability, that the structure is actually remarkably conserved. So on the right here, I have overlapped 1, 2, 3, 4, 5 CDR L1 loops from different antibodies. And these are differing in length. So the shortest one in dark blue is 10 residues long, up to, I believe, the longest one here in red, I believe, is 17 residues. So you can see that despite this variability in length and sequence, the structures are remarkably conserved, and all this variation in length is accommodated by this, what you might call rabbit ear, sticking up between this region of the structure. So his idea then was that you could define what he called canonical classes of loops. And obviously, if you've got a different length of loop, you have a different conformation. So clearly this 10 residue loop doesn't have the same overall conformation as this 17 residue loop, despite the fact that the overall shape is fairly similar. So he said that for each length, you can define a set of canonical classes, and which class a loop falls into will depend on the presence of certain, what he called, key residues. Now, at around the same time, I was doing my PhD and I was working on modelling of antibody structures, and we thought, well, okay, this idea seems to work for the 8 or so structures that were available at that time. But given that there are billions of different possible antibodies, surely it's not going to work in general. Well, we were proved wrong and Chodhia was proved right. This is the latest paper that he published on this subject. by Bisan al-Lazikani and co-workers, including Cyrus Choudhia. And Bisan actually was an undergraduate here at UCL. She was studying biochemistry, and she went off to do her PhD afterwards with Cyrus Choudhia. Now, this paper, published in 1997, shows the progress then. At that stage, there were about 50 antibody structures. And on the left here, we have three different canonical classes of CDR-L1s. And you can see in the top one here, we've got about 3 structures. They're all very similar to each other. The middle one, we have 2 structures very similar to each other. The bottom one, again, about 3, very similar to each other. In the middle bottom here, we've got CDR-L3s, and you can see that there are probably about 10 different structures here, all very, very similar, and their conformations are dependent on these conserved hydrogen bonds shown as the dotted lines holding bits of them together. At the top, we've got more CDR-L1s, longer ones this time, and again, you can see about 3 structures in the left-hand one of those 2 in the middle, very similar, and then a couple which are longer still on the right. Bottom right, we have CDR-H1s, all extremely similar to each other, nice cluster of conformations there, and above that we have a CDR-L3 of a different length from the one down at the bottom there. Now, this idea of key residues, this is looking again at CDR-L1. This is the shortest one that we saw in dark blue a couple of slides ago. This time I have shown you the backbone, the NC-alpha-CO backbone of the CDR in green. And I have shown you the side chains of the key residues within the CDR in red. But the key residues don't have to be in the CDR. They can also be outside. So these 2 that are shown in orange are in the framework and pack against that CDR. So these 3, when you space fill them, all touch each other and form like the centre of a sandwich where the bread is shown in either side in orange. And once you have this little core that holds the structure of the loop together, you can vary these other side chains that I've shown in grey as much as you like, but you'll still have the same overall conformation. So the nature of these key residues is actually variable. You don't have to have exactly the same amino acid, and that's shown here, where to get this particular class 1, as we call it, conformation for a 10 residue loop, this framework position L2 here has to be an isoleucine. The other framework position 71 here, as it says here, can be tyrosine or phenylalanine. Position 25 needs to be an alanine. 29 here can be valine, isoleucine or leucine, and 33 can be leucine or methionine. Now, I don't expect you to remember any of that, but the point is that some positions are 100% conserved, other positions are variable, but within classes. As you can see, 29, valine, isoleucine and leucine, as you should know, are all very similar amino acids. And tyrosine and phenylalanine are essentially the same, except tyrosine has the hydroxyl group at the top here, and phenylalanine doesn't. So, providing you conserve those features within those groups, you know that you'll have the same conformation of loop. This is another example. This is CDR-H2. And in this case, it shows you how a key residue outside the loop affects the conformation. So all you need to see from this is that when you have a short residue at this position, H71 outside the loop, alanine, leucine or threonine, then you get a conformation like this in red, where the top of the loop flips over to the left. When you have a longer residue here, H71 is an arginine, then it causes the conformation to flip. So the important thing to remember from this is that these residues outside the CDRs have an effect on the conformation, and we'll come back to that again later. It's important to note, however, that this canonical class model does not apply to CDRH3, and this is because you will recall that the VDJ splicing means that the D segment is the part that encodes CDRH3, and because of this huge variation in the number of D segments, the 6 reading frames in which it can encode the protein, and the extra bases that can be inserted either side, we just have too much variability in order to be able to assign these sorts of classes. So the take-home message about conformation of CDRs is that the conformation or canonical class of the CDRs is determined first of all by the length of the CDR, and second by the presence of key residues, both in and around the CDRs. So not just residues in the CDR loops themselves, but ones that pack against the loops. And in fact, other residues which are not directly interacting with the CDRs can have a smaller effect on the loop conformation as well. And you'll see why all this is particularly important from the point of view of drugs as we go on later in the lecture. So, why do antibodies make good drugs? What is special about antibodies that means that so many biotech companies and pharmaceutical companies are interested now in producing antibody-based drugs? Well, the idea of doing this was actually suggested as long ago as 1899 by Paul Ehrlich, who suggested the idea of what he called magic bullets. In other words, drugs that would go with great specificity and affinity to a particular site in the body which needed the drug, without having side effects and affecting the rest of the body. Well, this is exactly what antibodies do. They have this high affinity, they have this high specificity, and therefore there have been many, many attempts to use antibodies to do this. As I said early on, around a third of drugs in development are monoclonal antibodies, or based on monoclonal antibodies. And this number comes from a review in 2007. So it's over 10 years old now, but those numbers are still probably about right. The International Non-Proprietary Names Committee, which is part of the World Health Organization and is responsible for giving not brand names but non-proprietary names to all types of drugs, gets about, it varies, but around about 50 every 6 months that have to be named, and that represents approximately a quarter of all the drugs that they have to produce names for, and this is usually done around about phase 2 clinical trials. As of April 2018, there were 72 approved monoclonal antibody products in the US or Europe, and this information is available from the Antibody Society's approved antibody list. And I would encourage you to join the Antibody Society. It's free for students. You get 2 years free membership, and you can have a look and see how many approved monoclonal antibodies there are on the Antibody Society website. So there was a paper in 2015 saying that around 4 new antibody-based products are approved for use every year. In fact, in 2017 there were 13, not 4. So there's been a huge spike in the number of antibody-based drugs that are getting to the market. So why use them? Why are they so important? Well, we've already said high affinity and high specificity. But why are antibodies particularly good at this? Well, what I show on the right here is an antibody FV fragment again, the VH and VL domains. That is in comparison with a small drug-sized molecule. In fact, I think this is phosphocholine, but it's around about the same sort of size as a fairly small molecule drug. So you can see in terms of the binding area that an antibody has, which is around the top here, compared with that small molecule, the difference is really quite huge. Now, what does that mean? Well, that means that for a small molecule, it can only make a small number of interactions. And that means that it really, in order to optimize and maximize the number of interactions it makes, it needs to bind into a pocket. Now, the other important thing is where binding energy comes from. Now, if you think back to the lectures on protein energetics, you'll recall that the interactions between two proteins or two molecules of any type depend on electrostatics, hydrogen bonds, van der Waals packing, and hydrophobicity. Now, as I said in that lecture, hydrogen bonding is really not that different when you consider the two molecules by themselves interacting with water and the two molecules interacting with one another. Water is the consummate hydrogen bonder. It can hydrogen bond very easily to those molecules. So when you look at the overall difference in energy, the energy that our two individual molecules have when they're hydrogen bonding to water is about the same as the energy they have when they're binding to each other. Similarly with electrostatics, common table salt dissolves very well in water. If electrostatics was such an important factor in keeping things together, then salt wouldn't dissolve. It would want to retain that crystal lattice where all those electrostatic interactions are so good. And similarly, the van der Waals packing energy, well, water can pack just as effectively with those molecular surfaces as they can with one another. So really the driving force for the binding energy is the hydrophobicity, burying hydrophobic patches away from water. Now, that's not to say that electrostatics and hydrogen bonding and van der Waals contacts are not important. Indeed, the opposite is true. If you imagine a perfect hydrophobic match between two surfaces, which gives you lots and lots of binding energy, but then you have a mismatch of hydrogen bonding, well, that's hugely disfavourable, because those hydrogen bonding residues would much rather be hydrogen bonding with water instead. So you have to match up all those things, and those give you the specificity and allow the energy that comes from the hydrophobic interactions not to be wiped out by unfavourable other interactions. So if you're talking about trying to get good binding energy from a small molecule, you really need that small molecule to be quite hydrophobic. Antibodies, on the other hand, give you such a much larger, flatter, or potentially flatter interaction surface that they can form far more interactions. And that allows you to have far more specificity as well as high affinity. The fact that the interacting surface can be quite flat also allows you to target antigens or antigenic surfaces that a small molecule could not interact with, because the small molecule needs a pocket, whereas the antibody doesn't. So this opens up a whole range of protein targets that antibodies can bind to that small molecule drugs cannot. How then can we use these antibody-based drugs? What mechanisms can we use to exploit this high specificity and high affinity? If we talk about ways in which we can use antibodies in vivo, well, we can use them for imaging. We can use them as therapeutics, and that's really what I'm going to focus on. And we can use them kind of indirectly as therapeutics for targeting other things to a site that we're interested in. So, talking about antibodies as drugs then, there are really 4 ways in which we can use them. We can use them in the same way that we use conventional drugs. In other words, all we're interested in is their ability to bind things, because that's what a normal drug does. Usually, with the exception of some anti-cancer drugs which bind to DNA, but other normal drugs bind to proteins. And there they can block interactions in one way or another. So they can act as enzyme inhibitors. In principle, they can also act as allosteric activators of enzymes. If they're binding to a ligand of a receptor, they could block the interactions between that ligand and receptor. Now, with a small molecule drug, that's not normally something you can do, because ligands, such as hormones, tend to be fairly small and convex in shape. And small molecule drugs, which need a pocket into which they're going to bind, can't bind to these things very effectively. On the other hand, antibodies can do that really well. If the target to which a drug is binding is a receptor, then just as with small molecule drugs, they can fall into 2 categories, either agonists, which switch the receptor on, have the same effect as the normal ligand binding, or antagonists, which prevent the receptor from being switched on. The second way in which antibodies can work is to trigger the rest of the immune system to do something. So that first method, as conventional drugs, is just involving binding. The second method requires the Fc fragment of the antibody to trigger things like complement-dependent cytotoxicity. This is the complement system, which is designed for killing cells, or antibody-dependent cell-mediated cytotoxicity, or ADCC, which happens via the various types of receptors for Fc fragments that we talked about in the last lecture. The third way in which antibodies can be used as drugs is for targeting or delivery. So we can have a radioactive payload, in other words, we have some sort of chelating agent attached to the antibody, and that then holds a radioactive ion in place. The antibody goes to the site of interest and the radioactivity kills the surrounding cells. Second, we can use a chemical payload. This is what's known as an antibody drug conjugate, or ADC. And these drugs are often coupled via a linker which allows them to be released in certain circumstances. So tumours tend to have a slightly lower pH than the rest of the body. And so these linkers can be made to be pH dependent and release their chemical drug payload when they strike the lower pH. Alternatively, the antibodies can be internalized by cells where they're broken up and again the drug payload is released. And then thirdly, we have techniques for indirect delivery, and in particular a technique called antibody-directed enzyme prodrug therapy, or ADEPT, which we'll come back to in just a minute. The fourth way in which antibodies can work as drugs is by cross-linking things. And to do that, we can create so-called bispecific, or indeed these days trispecific antibodies, which have multiple binding sites for different ligands and therefore can be used to bring things together. For example, bringing together a T cell and a cancer cell, such that the T cell will kill the cancer cell. In the context of being interested in bispecific or trispecific antibodies that can cross-link different things, and also in the context of using antibodies as drugs that bind, where we're not interested in triggering the rest of the immune system, then we don't need the Fc fragment down here, because that's the region that triggers the rest of the immune system. So in these cases, all we actually need is this Fab fragment, the antigen binding fragment. And so people have produced antibody-based drugs that actually consist only of this Fab fragment. In fact, of course, all we really need is the FV, the variable fragment, consisting of the VH and VL domains, because that's where all the variability is, and where the binding site is up the end here, which interacts with the antigen. So Fab fragments are fine. The problem with using FVs is that they're not very stable. The two chains tend to fall apart. So what people have done is to create so-called DSFVs, disulfide FVs, where an additional disulfide bond is introduced which links the two domains together. Now this makes a very stable FV fragment. The problem is that the yield of these is not particularly good, because remember that both the light chain and heavy chain contain a disulfide that holds the two sheets together. So as the protein folds, if there's an extra cysteine in each chain, then you can get scrambled disulfides. So when you do get correct protein forming, it's very good and very stable, but the yield of that protein is not so good. The second thing you can do is take the FV region and artificially link either the C terminus of the heavy chain to the N terminus of the light chain, or vice versa, the C terminus of the light chain to the N terminus of the heavy chain. And I've shown here, I've just sketched in as a red line what this linker would look like, because these molecules, the linker tends to be quite flexible. It's usually made up of glycines and serines, designed to be flexible, not to put any constraints on the structure. But this is enough to hold the two chains together and keep them from just separating and falling apart. So single chain FVs are another way forward for producing something that may be useful as a drug when you only need to bind. It's also useful for something else, which we'll come back to in just a couple of minutes. In practice, however, it's unusual to use anything smaller than a Fab, and often people do actually just use intact antibodies. And the reason for that is that things like Fabs and smaller proteins tend to get cleared from the bloodstream rather quickly. They just get turned over rather rapidly. The bigger the molecule is, the slower it gets turned over. And people have actually solved that in the case of Fab fragments by pegylating those, adding polyethylene glycol, and that just increases the size of the molecule and gives it a longer half-life. Now, one of the examples of how we can produce something that can cross-link things is actually exploiting this idea of a single chain FV. So what we're doing with the single chain FV up here, this red region of the IgG antibody, as I showed you before, all we want to do is link the N terminus of one of the chains to the C terminus of the other. Now, if you make that linker rather shorter, then instead of forming a single chain Fe, what you get is two of these pairing up. So you get the VL of one of the pairs joining with the VH of the other. And what you end up with as a result of that is a bivalent antibody. In other words, one which has two binding sites that bind to the same thing. Now, if you play around a little bit with the DNA that encodes this, such that you have the VH of one antibody followed by the VL of another, and then the VH of the second antibody followed by the VL of the first antibody, and these are two separate genes that you're providing, you end up producing something that looks like this. So the VL and VH of one pair come together, and the VL and VH of the other pair come together. And this way you end up with your bispecific antibody formed from two single chain Fes with the ability to cross-link things. Now, this is an example of a bivalent antibody. It's an antibody called T8466. The other name for these, as it shows here, is diabody. This happens to bind to CEA, which is carcinoembryonic antigen, which is overexpressed in colorectal cancer. And so you end up here with one binding site up this end, one binding site down this end, and you can see the linkers here and here. Now, being able to do this sort of thing, to be able to produce different types of antibody with different valencies, different numbers of binding sites, different ability to bind to different things, has started to be exploited very widely. So top left here we have a standard IgG antibody. Here we have one where the CH2 domains have been removed. Here we've got a Fab prime. Here we've got what's called a mini body. We've taken out the CH1 and CL domains and just got the CH3s. Here's a Fab. We have the single chain Fes, as we've seen. We could just have a VL by itself if that has enough binding ability. We just have a VH by itself. And this is sometimes known as a mini body because, in fact, camels or camelids, so llamas as well, actually have ordinary antibodies, but they also have antibodies that are formed only of heavy chains. So we can exploit the kind of surface of the heavy chain that would normally be interacting with a light chain from a camel or a llama, because those surfaces are slightly different, because they're not as hydrophobic, so they don't tend to form these dimers. So we can camelize the surface of an antibody that we're interested in so that we get just a VH domain. And, you know, all these different formats were being considered, things with artificial linkers shown in red, bottom left here. All these different formats were being considered in 2006. Now, this slide comes from 2015, and I'm not going to go through any of these, but we're now ending up with a huge range of different bispecific immunoglobulins on the left here. Bispecific antibody fragments over here, appended IgGs, so this is like a normal IgG antibody with extra binding ability stuck onto it. All sorts of things being produced, and this is in 2015. And the range of such molecules is only going to increase. There are lots of these sorts of things in development at the moment. As far as I know, none of them are approved for use as drugs, but certainly there are lots and lots in clinical trials currently. Some example applications of antibodies in a little bit more detail. In vitro antibodies are invaluable. They can be used as lab research agents for purification and for labelling and for diagnostics. In vivo, they can also be used for labelling and for imaging, and as we've already seen, they can be used as drugs. So let's just look at a few examples of these sorts of things, many of which you'll be aware of already. So they can be used for purification. As I said, you put them, the antibody on a column, you run your mixture of proteins down the column, protein of interest will stick, you wash off everything else, and then you elute your protein of interest. They can be used for labelling in things like Western blots and ELISAs. And there are many companies where you can purchase antibodies off the shelf. There are several companies also that will produce antibodies to order. So you provide them with some pure antigen and they will make antibodies for you. The sensitivity and specificity of antibodies allows the detection of very low concentrations of very specific antigens. So basically what you're doing is just using them to detect the presence of an antigen of interest. And there are many, many monoclonal antibodies in use for doing this. Things based around ELISA type assays. Interestingly, sheep antibodies are proving quite useful in this sort of area. So there's actually a company called Bivalent in Farnham in Surrey, which specialises in producing sheep antibodies. And it seems that sheep are able to produce antibodies that are specific with high affinity for certain molecules that we can't produce high affinity specific antibodies for in more conventional animals like rats or mice. It's not really clear why that is, apart from sheep presumably being, well, obviously being larger animals, but presumably being more developed animals and having a more developed immune system. In vivo, we can use antibodies for diagnostics and imaging. So a technique called radioimmunoscintigraphy, in which radio-labeled Fabs can be imaged. And this has been used for this carcinoembryonic antigen, again, that I mentioned for colorectal cancer. So basically, by giving the labeled Fabs, imaging where they appear, you can see whether the tumour has spread. It's also been used for neuroblastoma. In practice, those techniques tend not to be used so much these days because conventional imaging techniques such as CT scans and MRI scans have become so much more sensitive that there's less need for these sorts of more invasive techniques. There have also been experiments with what's known as radioimmunoguided surgery, in which a patient is injected with radio-labeled antibody before surgery. This then concentrates at the site where tumours are found, and then once the surgeon opens up the patient, then using a handheld gamma probe, they can find sites of radioactivity which will indicate metastases of the cancer, so typically to the liver or to the lungs. And again, anti-CEA antibodies have been used in this way for colorectal cancer. So I've mentioned cancer quite a lot, and cancer really is one of the main targets for antibody-based treatments. This is just a small range of antibodies with different antigens shown in the third column. Just to point out here, I've used the commercial name here on the left. So this is the name a drug company gives to its product. The INN is this international non-proprietary name. So once a drug goes off patent, and that means that other companies can then start to produce copies of the drug, they can sell it under this name. And they can make up their own commercial names as well, but this name allows doctors to see that this is the same thing as Erbitux. If it's prescribed as cetuximab, then the chemist, the pharmacist who is actually giving the medicine knows that they can give Erbitux or they can give whatever copy of the drug they like. So you can see a range of different cancers that have been targeted. Early antibodies really were only suitable for blood cancers because the antibody molecules are not so good at penetrating into solid tumours. But some of these, such as this last one here, Avastin, this actually binds to VEGF, vascular endothelial growth factor, which is involved in vascularization of the tumours. So basically, by blocking VEGF, it stops the blood vessels from forming around a tumour, allowing the tumour to grow. So by stopping that, the tumour can't grow anymore and makes it easier to attack with other drugs. The three mentioned just before here target programmed death receptor 1 or programmed death ligand 1 or CTLA4. Now these are all interesting antigens that are involved in tumours being able to escape the immune system. So by blocking these molecules, the immune system is more able to kill cancer cells. This is probably the best known of the anti-cancer antibodies. Commercial name is Herceptin. The INN is trastuzumab. And this binds to HER2, which is a specific EGF receptor, which is overexpressed in certain breast cancers. Now, patients who have this HER2 being overexpressed tend to have a worse clinical outcome and worse prognosis than people who don't have HER2 positive breast cancers. But with trastuzumab, then the prognosis is at least as good as in patients who don't have HER2 positive breast cancers. Now, this was quite an old news story from 2005 when this lady, Elaine Barber, was refused Herceptin by her local primary care trust because of the expense of these antibody-based drugs. And this is one of the main problems, that manufacturing these things is generally a lot more expensive than manufacturing small molecule drugs, because the process involved in producing a biologic and ensuring that that biologic is consistent and pure, has the same glycosylation and all these sorts of things, becomes more expensive than something that you can do in a chemistry lab. So Elaine Barber, who was refused this molecule, actually went to the High Court and had this decision overturned and was eventually prescribed Herceptin or trastuzumab. So a lot of the use of antibody-based drugs is for cancer, just because cancer is so difficult to treat that you can kind of justify using such expensive types of treatment. But in fact, there are huge ranges of diseases these days that can be treated using antibody-based drugs. Psoriasis, osteoporosis, migraine, HIV, haemophilia, asthma, multiple sclerosis, rheumatoid arthritis, high cholesterol, Crohn's disease, anthrax, macular degeneration, and so on. Rheumatoid arthritis, I mentioned the world's top-selling drug, which is adalimumab. The commercial name is Humira. And this is actually a treatment for rheumatoid arthritis and related conditions. But here I've just given a small sample of the sorts of things that are used. This one, Xolair or amalizumab, actually targets IgE antibodies. So this is an antibody being used as a drug that targets other antibodies. And this is used in severe allergic asthma. So probably around about 2010, 2012, something like that, there was a story in the news about a boy who suffered from severe allergic asthma. It was so bad that basically, he couldn't go out of the house at all because of being exposed to pollen and other allergens. Given this drug Xolair, he was able to go out, play football with his friends, and was able to lead a normal life. And this came up in the news because NICE, who regulate what the NHS are going to pay for, had decided that Xolair was not commercially viable. It didn't give a good enough return for the cost. But obviously in some cases, at least for this particular child, it was revolutionary in what it did. And I think NICE has now reversed that decision. Another story that you might have heard about some time ago now, in 2006, was an antibody designed to modulate the immune system. Now, actually, quite a lot of antibodies are doing this. So the ones that I mentioned earlier that bind to PD-1 or PD-L1 or CTLA-4, although they're being used for treating cancer, they're described as immunomodulatory antibodies because they are interfering with the suppression of the immune system. Now, in 2006, there was a drug trial looking at this antibody called TGN-1412, which bound to CD28. Now, if you recall from the previous lecture when I did my one-slide summary of immunology, there's this interaction between an antigen-presenting cell, which has MHC on its surface with a peptide, an antigenic peptide bound to that MHC, and that interacts with a T cell receptor on a T cell, and that's shown in this top image here. Now, if that happens alone, we get partial activation of the T cell, and actually we build up tolerance against that antigenic peptide. However, if that happens in concert with another interaction between a molecule called CD8086 on the antigen presenting cell and CD28 on the T cell, then that leads to full activation of the T cells and priming against seeing this antigen again. Now, what TGN-1412 was designed to do was to bind to CD28 and activate T cells without this MHC restriction happening. And the drug company called Tegenero, who were doing this trial, said this had possible applications in leukemia and in arthritis. Now, in leukemia, you would probably be wanting to boost the immune system. In arthritis, you'd be wanting to damp down the immune system. So it's not really clear that they knew what they thought this would be useful for. But they had done lots of trials, including in cynomolgus monkey, and they had a dose that seemed to be very safe. Turned out when they gave this drug to human patients or human volunteers, in fact they were healthy volunteers, or they were before they had the drug, that it led to what's known as a cytokine storm. They activated all the T cells, and these T cells started to release cytokines which started to attack their body. Some of the volunteers lost fingers, one's head swelled up to twice its normal size. Really quite horrific side effects. Fortunately, all of them survived, but actually only just. And it turned out that even though cynomolgus monkey and humans are so closely related, cynomolgus monkeys and humans had slightly different populations of T cells, and this was activating T repressor cells far more in the monkeys, whereas it was activating the killer T cells far more in humans. Here are a few examples of antibodies that have been used for delivering drugs. So Bexar or tositumumab I-131, this is an example of an antibody with a radioactive label, radioactive iodine in this case, targeting CD20 in order to kill B cells in patients with non-Hodgkin's lymphoma. This is an example again of an antibody binding to CD20, this time with this is an ADC, so it's a drug that's conjugated to the antibody and again kills B cells in lymphoma. This one, this time it targets CD33. It's used in acute myeloid leukemia and again is an ADC where you've got ozogamicin as the drug molecule coupled to gemtuzumab. And this one's an interesting one, which was licensed, as I recall, early in 2018 and approved by NICE for use under NHS prescribing. This is trastuzumab again that we've already heard about with that lady Elaine Barber, but this time it's an ADC. An antibody drug conjugate with a conjugate called emtansine. So it has all the effects that normal trastuzumab has, but it also delivers a cytotoxic drug. In this case, they've actually added the word ADO onto the front of the INN name to help avoid prescribing errors, because they were worried that if somebody just wrote trastuzumab emtansine on a prescription, the emtansine bit might get forgotten about and the patient would just be given normal trastuzumab. And this has the commercial name of Kadcyla. Earlier, I also mentioned this idea of indirect drug delivery through a process known as ADEPT, or antibody-directed enzyme prodrug therapy. Now, what we do here is take an antibody and conjugate it to an enzyme. We inject that antibody enzyme conjugate into the patient and it localizes at the tumour cell. The antibody binds to the tumour cells. Now we inject a prodrug. Now, a prodrug is an inactive form of a drug which usually has some little extra bit added to it. In fact, many drugs that we take are in fact prodrugs. So the painkiller codeine is actually a prodrug. It gets activated in the liver into the active form. Now, what happens here is that this enzyme is designed to break this prodrug down and release the active drug and some sort of inactive leaving group. So the idea here is that you produce a high concentration then of the drug just where the tumour is and not around the rest of the body. In practice, however, ADEPT really doesn't work very well at all. The high concentration effect is minimal and consequently you have to use extremely toxic drugs, and those then just kind of leak away, spread around the blood system and just spread their toxicity everywhere else. There are also potential severe immunogenicity issues because if we use if the enzyme part is not human, then the human host will recognize this as non-human and will launch an attack against that enzyme, so that when you give it a second time, it really just doesn't work very well. We'll come back to antigenicity of the antibody itself in just a moment, but if there is any antigenicity in that antibody, then again we can attack that, recognize that as a foreign protein. The immune system attacks it. that would have the same effect as the attacking the enzyme. But if we had chosen a human enzyme to use, which might be sensible because it would reduce the chance of immune attack, if we did get an immune attack onto the antibody part, we can get a sort of immunological drift, such that we have antibodies that bind to the antibody part, and then we can start to produce more antibodies that react to the enzyme part. And because this is a human enzyme, we then start to attack that human enzyme throughout the body, and that obviously can have fatal consequences. So that leads us very neatly onto the problems of using antibody-based drugs and solutions to those problems. When we produce monoclonal antibodies in the conventional way, as I mentioned in the last lecture, that's done usually by introducing the antigen into mice or rats or whatever, harvesting B cells that produce the antibodies, and then fusing those B cells with myeloma cancer cells. This forms what's known as a hybridoma, and this is immortal. It carries on dividing and producing large amounts of antibody. However, this process doesn't work with human B cells. Nobody has managed to find a cancer cell line that will fuse properly with a human B cell. Also, of course, most of the targets that we're interested in are actually human proteins, and the human immune system doesn't target human proteins. If it did, we'd all be suffering from autoimmune diseases. And if you wanted to target a non-human protein, then that's probably on some rather nasty bacterium or virus or something, and you probably don't want to introduce that nasty antigen. It might be a toxin. So obviously there are people who have survived nasty infections, HIV or Zika virus or whatever it might be, and you can harvest antibodies from those people and then clone them and make them artificially, but you can't do it by the standard monoclonal technique. So what people tend to do is use mouse antibodies or rat antibodies, because those can be produced very easily. The problem is, when you take a mouse antibody and you inject it into a human host, that will lead to an immune response, not in the sense of the antibodies switching on the immune system to kill something, but in the sense of our own immune systems recognizing this antibody drug as a foreign protein and producing an attack against that drug. And this is known as the human anti-mouse antibody response, or HAMA response, or more generally as an anti-antibody response, or AAR. So people came up with ways of trying to get around this problem. And the first of those techniques was to use what are known as chimeric human antibodies. So in this case, what you do is you produce your antibody in mice, and then once you've got the sequence of that antibody, you can produce artificially DNA which encodes the light chain of the light chain variable region of a mouse antibody coupled to the human constant region, which we show in yellow here. And similarly in the heavy chain, we take the VH domain from the mouse and we couple it to the constant domains from a human. Now, this is obviously far less mouse protein present and more human protein present, and therefore it's less likely to induce an immune response. It also has the advantage that the FC fragment here is the human FC fragment, and therefore it's better able to trigger the effects of ADCC and CDC that we saw earlier in order to kill targets. Raguska and co-workers in 1994 also had a slightly improved version of this, where they looked at the surface of the VH and VL domains and said, are there any residues on the surface away from the binding site which are unusual in human antibodies? And if so, we'll just change those to the human residues instead. And that will give us a chimeric supporting the loops in the way that the mouse loops were supported, but with a surface that looks like a human antibody, and therefore hopefully reducing the immunogenicity further. However, it was realized that actually it is just those CDR loops that form the binding site. And this is that antigen's eye view of the binding site again. This time the binding site is shown here. It's the purple, orange, yellow, green, red and cyan loops that are forming the binding site. There are these two other loops which don't form part of the binding site, which are shown in pink, and then the rest of the framework is shown in blue and the kind of greyish green. You can see a couple of the framework residues poking up through into the binding site, but essentially it is just these loops that form that binding site. So what we can do again with engineering at the DNA level is produce a hybrid where everything is human except for those CDR loops. And this was first done for an antibody that bound to a protein by Reichman et al. In 1988 in this paper in Nature called Reshaping Human Antibodies for Therapy. They actually used a rat antibody, and they grafted the CDRs onto a human antibody called REI, which was used as a framework to support the light chain CDRs, and an antibody called NEW, which is a human antibody which was used to support the heavy chain CDRs. So the two heavy chains came from NEW, the two light chains came from REI, the CDRs came from this rat antibody. However, they found that when they did this, they didn't get such good binding to the antigen. Now, Reichman was working in Greg Winter's group, and as some of you may know, Greg Winter actually won a Nobel Prize in 2018 for some of his work related to antibodies. Actually, not this bit of work, but another bit that I'll mention later. So Greg Winter's group was in Cambridge. Now, Cyrus Chodhia, who I mentioned earlier, who did this analysis of the conformation of these antibody CDR loops, was also working in Cambridge. And from Chodhia's analysis, you will recall that the conformations of the CDRs are determined by these key residues, some of which are within the CDRs themselves and some of which are outside the CDRs, and that certain other residues outside the CDRs can have an effect on the precise conformation of the CDR loops. So Greg Winter looked at the structure of this particular antibody and found that CDRH1, which is shown in red here on the right, packed against this bit of structure in the framework, which is shown in green and orange. Now, this residue, phenylalanine, was packing against this tyrosine in the loop. However, when he replaced this rat framework with the human framework, there wasn't a phenylalanine here anymore. Instead, there was a serine. And serine, of course, has a much shorter side chain. It would end just where the ring of the phenylalanine starts. So he thought, if I mutate this serine to a phenylalanine, I should restore the packing and restore the conformation of CDRH1 over here in red. Now, he also looked at another mutation at position 30 up here, where the rat antibody had a threonine and the human framework had a serine. He tried making that change as well. The serine to phenylalanine mutation completely restored the binding. The serine to threonine mutation had really no significant effect. Now, it's important to note that this is a specific example. It's dependent on the packing of this tyrosine in this particular rat antibody against phenylalanine that was present in the framework of this particular rat antibody. Now, not every rat antibody will have a phenylalanine here, and not every loop will have a tyrosine here. But the concept remains. In other words, that certain particular positions may be important in supporting the CDRs in precisely the correct conformation that's needed for binding. And of course, this is commercially hugely interesting. And there are a number of patents now related to how to find which residues you should change. So, for example, the Adair patent, Adair is the name of the inventor, specifies these 4 particular positions in the heavy chain. H6, which is here at the bottom, which packs inwards towards the strands that support the CDRs. H50 up here in yellow, again at the base of one of the CDRs. H24 in here in blue, which again packs inwards towards the CDRs, and this position H23, which actually sticks outwards. So this is the antigen up at the top. The CDRs for the heavy chain are shown in the thicker lines here in green. This residue really sticks outwards and has no obvious role. However, it was found in this patent that it had a 10-fold increase in binding when they changed this. I believe it was from a serine to a lysine, as shown here, had a 10-fold influence on binding. And presumably that occurs through this additional loop, the one that I showed in pink in an earlier slide, which doesn't form part of the binding site. But this residue presumably interacts with residues in that, maybe alters its conformation slightly, and that then packs against the CDRs and modifies their conformation. So a kind of relayed effect. So we've gone through this scale of starting with a fully mouse antibody, going through a chimeric, where we've replaced all but the FV region with human antibody, through to humanized, where we've replaced everything except the CDRs, plus a few extra residues to support the CDRs in precisely the correct conformation. And of course, the ultimate is to produce fully human antibodies. And the idea, as you proceed along this scale, is that you're going to decrease the chance of this antibody-based drug from raising an immune response against the drug. So why is this such a big problem? Well, in the best case, giving one of these drugs, you might produce an immune response against it, which simply means that the next time you give the drug, it'll be wiped out from the system very quickly and the drug will have no effect. In the worst case scenario, it could lead to anaphylactic shock and even death. Now, what I've shown at the bottom of each of these is part of the name of these international non-proprietary names. So mouse antibodies were known as OMAB, chimeric Iximab or Zimab, humanized Zumab and human UMAB. Since April 2017, those names have no longer been used because although there's an assumption that a human antibody will be less immunogenic than a humanized antibody, should be less than a chimeric and so on, less than a mouse, although on average that is probably true, it's not necessarily true for any particular individual antibody. For example, for those two antibodies that I've talked about quite a bit, Herceptin or trastuzumab is humanized, while Humira or adalimumab is fully human. Yet actually, more people have an immune response to adalimumab than they do to trastuzumab. So, although on average this is true, that you get decreased immunogenicity, in particular instances it may not be true. And so people were effectively trying to abuse the naming system just to get an UMAB name or a Zumab name rather than a Zimab name, just for marketing purposes. And so this was one of the reasons that it was taken out of the naming scheme. Just for the last few minutes, I'm going to talk about a couple of methods that can be used to produce fully human antibodies. And the first of these is phage display libraries. And this is the area in which in 2018 Greg Winter and George P. Smith shared half of the Nobel Prize for their work in developing this technique. So what we do here, with antibodies at least, is that we produce single chain FVs, which I talked about earlier, and we have DNA that encodes a whole library of millions of different single chain FVs. And that library of DNA is inserted into bacteriophage. So each bacteriophage has the DNA to produce one particular antibody. And that antibody protein is coupled to one of the coat proteins on the phage. So you can see here in red, grey and white, we have these little single chain FVs stuck onto the phage. So we immobilize some of the antigen of interest on a plate, we add the phage, and then we wash off what doesn't stick. We then elute the antibodies and phage that do stick, and we can stock and titrate these, but then we can also amplify them so that we've got more phage with more antibodies that bind. And we can go around this cycle 3 or 4 times and produce a whole set of antibodies that really bind extremely well to our antigen. One of the other things that we can do during that process is during the amplification step, we can allow mutations. So we can use a low stringency copying such that mutations are introduced randomly. And that allows us then to reproduce the effects of somatic hypermutation in selecting antibodies that bind really well. So we start that process using a library of antibodies, typically just normal human antibodies. But it could also be a library of antibodies from a patient who has been exposed to a particular virus, and therefore you're getting what's known as an immune library. The alternative technique is to use transgenic mice, so-called xenomice. And what's been done with these mice is that the gene segments for the variable domains, so the V's and J's of the light chain, the V, D and J for the heavy chain, have been removed or silenced and replaced with the equivalent human gene segments. And this means that those mice produce fully human V domains when challenged with an antigen including a human protein. So you give them human CD20 or whatever it is you're interested in producing an immune response against, or producing an antibody against, and those mice will recognize this as being a foreign protein, but they will produce fully human antibodies. The V domains are human, but of course the constant domains, you haven't tampered with those, so those are still mouse constant domains. And once we have those, we can again engineer those in the lab in the same way that we would with a chimeric, in order to produce an antibody which has human constant domains as well as human variable domains. And this method has been a very successful method over the last 10 years or so of producing fully human antibodies. It was limited to start with by the fact that the intellectual property surrounding this approach was very strictly guarded, and there were only one or two companies producing these things, and they charged huge amounts of money for them. Now I believe they have all gone off patent, or at least they're licensed on the basis that they're not quite as ridiculously expensive for companies to use, and therefore they're becoming a very, very popular way of producing fully human antibodies. In conclusion, we have examined CDR conformation in a little bit more detail, and the fact that for at least 5 of the 6 CDRs, the conformation falls into particular so-called canonical classes, and those classes are influenced by key residues present in the CDRs and residues that pack against the CDRs. There are also residues that really kind of fine-tune the precise conformation of the CDRs, and that became important later on when people were trying to humanize antibodies by taking mouse CDRs, grafting them onto human frameworks, and needing to make a few changes in those human frameworks in order to support the CDRs in precisely the correct conformation. We described some of the applications of antibodies both in vitro and in vivo. We looked at artificial antibody fragments, in particular based around Fabs and single chain FVs, but also mentioned this huge plethora of different formats that people are starting to produce based on antibodies with multiple specificities in order to be able to cross-link things. We examined the problems of using antibodies in vivo, which all surround immunogenicity, the fact that when you take an antibody that is not human and inject it into a human host, you will produce an immune response against those antibodies, and then solutions to that problem by producing chimerics, by doing humanization, or by producing fully human antibodies. However, I also made the point that even a human antibody can elicit an immune response in particular individuals, and sometimes a chimeric or a humanized antibody will induce less of an immune response. And finally, the LESC course textbook, edition 3, pages 265 to 268, has some more background information on this.